So hopefully you will have your models in front of you that you're going to be making, and I'll let Melanie and Dominic give you some more information.
Right, thank you very much, and thank you for inviting us to do this workshop with you today. We wish we could see you. That's the one weird thing for us. So throughout the workshop, there may be points where we ask you to give us a thumbs up. Do you think we can maybe have a quick try of that? Can everyone give us a quick thumbs up so we can see where you're at? Can you do that for us?
Yep, that's it. They're coming through.
Okay, perfect.
Right.
Wonderful. Okay, can we have the next slide, please?
So we had the inevitable technical issue, so apologies for this. So we can go to the next slide.
Fantastic. So we'll just start a little bit about us as people from Origami Organelles. We're both former research scientists, and we'll give a little bit of a background to each of us in a minute. And then we moved from research science to working into science education for the last 20 years or so.
Yeah.
And Origami Organelles is our own company. So from my background, I'm originally from Canada, and I did my first degree in genetics at the University of British Columbia in Vancouver. And I came to the UK and did a PhD in immunology at the University of Wales College of Medicine. I then worked at the University of Oxford for a couple of years in a different area of immunology. But during that time, I was always really interested in sharing what I did in the lab with students in schools because I really love science. And that led me into a whole other career working in biotech companies, teaching teachers and students how to do DNA technology in the classroom. And that's, I'll pass over to you now.
Okay, so yeah, I'm Dominic. So I did a degree in biology, and then I did a PhD in developmental biology, so looking at the genetics of how embryos develop in the womb, working on zebrafish. And then I did a little bit of postdoc, and then I worked for a biotech company, an American biotech company called Bio-Rad, and ran their education program for 6 years across Europe, and worked for another American company after that, and then for 7 years, and then we've set up our own thing since then. And along the line, we had this idea for making paper models to teach science, and it's become quite a big thing now, and especially with the current coronavirus situation, which we're all kind of well aware of, it's been become very interesting for a lot of people. So we're going to make one of those models today.
All right. One thing just to carry on just a career thing a little bit is in terms of Origami Organelles, we have been always working from home, so that part of the situation isn't strange for us. But what's interesting to maybe know is, you know, we're based in Hartford, and we sell our Origami Organelles all over the world. So it's been fantastic. The only continent is not that we haven't sold to is Antarctica, so that's fun.
We're hoping they'll get something sometime.
All right, can we have the next slide, please? All right. We don't have to tell you what's happening in the world today about coronavirus affecting all of us. But what today's workshop is about the science behind coronavirus. And we're going to be working on the making a model of the structure of it to talk about some of the implications that structure has on things like testing and vaccines. So that's what we're going to do today. Next slide, please. Okay, so we're going to begin with, just because we don't know exactly how much everybody knows about viruses, we're just going to talk briefly about what a virus actually is. Now, a virus is a small particle that invades cells. So I like to think of them as being like pirates that take over a cell. And they aren't living things because they can't replicate or reproduce on their own. One of the real difficulties for treating viruses is they're difficult to treat, unlike bacteria, because they hide inside cells. So that's one of the big problems with viruses and why there isn't a cure for a virus. Next slide, please. Now, the coronavirus pandemic that's affecting all of us is a new virus, and the coronavirus, the disease that is caused by this new coronavirus is called COVID-19. So COVID-19, as most of you know, causes severe pneumonia that sometimes can be fatal in older people and those with pre-existing medical conditions. And it first emerged in China in December 2019 and is thought to have transferred to humans from bats. Now, as I just said, viruses can't be treated by antibiotics, and there isn't yet a vaccine for this, which would help our immune systems clear it up. So we're going to talk about that in a little bit more detail. Next slide, please. The biggest difference about coronavirus, between coronavirus and other viruses that we get infected with every year or all the time, are that coronavirus can infect people and they can be asymptomatic. And asymptomatic means they don't have any symptoms, so they won't even know they have the virus. So they therefore spread the virus without knowing it. And last week in the news, Cristiano Ronaldo has tested positive for coronavirus, and he is completely well. And because in the footballing world, they're tested a couple of times a week, that's how he found out. So that's the problem with coronavirus and why we are having all the restrictions we are having. Next slide, please. Okay, just to briefly, again, talk about how viruses work. What we have in the top corner there with the arrow pointing at a virus, is a little particle that will come in and attach itself to the surface of a cell. And it attaches to a protein on the surface of the cell with a protein on the receptor, on the virus. Now, this is not the normal thing that would attach to the protein on the cell on the little green blob, but the virus has hijacked it. And what it does then is that it fuses into the cell and uses some of the cell's own machinery to copy its own genetic material to make lots and lots of new baby viruses that will then burst the cell, and that's what causes the illness, and then release lots more viruses to infect neighboring cells and to be, for example, coughed out of a person and infect other people. All right, next slide, please. So we're going to look at the structure of the coronavirus that causes COVID-19. It's got several features. We've got the proteins around the surface of it that we'll talk about in more detail as we go on, the genetic material that's inside, and some proteins as well. Next slide, please. Okay.
All right.
We're going to make our models. Hopefully everyone has them already cut out. And I think you can probably still see us in the corner. Dominic's going to take you through the making of the model.
You, I don't know how much of this you can see if I hold it up, but that's hopefully what you've got. So we're going to start with this piece. So if we could go to the next slide, please. Okay, you can skip through that one. Okay, right, so this is the first part we're going to make, which is called the envelopes. This is the outer membrane around the virus. So hopefully you've had a chance to cut it out, cutting around all the twiddly little bits on the edges as well. And then we're going to show you how to turn that into a virus in a second. So if you could go to the next slide, please. So one of the things it's possible to miss is just there's these tiny little slits which need to be created. So you just need to cut, sorry, there we go. You need to cut these little lines here as it shows on the slide with a pair of scissors. I'm going to do mine now quickly, but I'm not sure if you can see what I'm doing.
And you just want to make sure you don't cut all the way through the piece.
And one of the nice things about paper models, obviously, if you do cut through it, you can just tape it back together. So it's not a disaster if you do, but try not to. Okay, so those little, if you go to the next slide, please. So the next thing you need to do is just fold up the sides along the dashed lines. So just fold these up so they're sitting as shown in this photograph. And then next slide, please. And then we're going to just fold them together. So this is when we use those slits. So if you just curl the membranes over, so try not to cover that up with my hands while I'm showing you. And then if you just slot those together, thus, and then you just kind of push them together. You can, if you have tape, if you want to use it, you can use that to hold them together more firmly, but then those should join together quite neatly. If you go to the next slide, and then fold them down, so as shown. So that's what I've done here. And if you just do that to the same on the other side, if you could, next slide, please. And then if you repeat at the other end, you end up with this shape, circle with the sides joined together.
All right, why don't we just give everyone a minute, and if you could just give us a quick thumbs up to let us know that you're okay with all of that, that would be wonderful.
Should we scroll down?
Mm-hmm.
Okay, we'll just give you guys a moment or two.
This would be a good idea for you to use the chat function and see if it works for you. So let us know if you're ready with using the chat function. Thank you.
Oh, I see some thumbs. there's no need to rush. Don't feel pressurized. We'll wait for you all to do it.
And if any questions, you'll let us know, will you? Helen, is that okay?
Yep, so we'll let you know. We'll let you know
Okay,
if any
thanks.
questions come through. There's none yet. But if anybody does have any questions, please feel free to put them in the chat and we'll ask Melanie and Dominic.
If we keep that. Okay.
Yeah.
Okay. Shall we move on?
Should we carry on? Okay, so anyway, you've got the part. So there we go. So that's where we were up to. If we go to the next slide, please. Okay, so what we're going to do now is just fold down all those sticky out bits, and these will explain in more detail. So just fold them down, as you can see in the picture. It's probably easier for you to see it in the picture than what I'm doing. And then these are now on the outside of the virus. So these are the membrane proteins that Melanie mentioned earlier on. So we now have, and this is why it's called coronavirus, because it has this whole arrangement of things on the outside and makes it look like a crown. So that's where the name comes from. So if you just fold those down, and then we'll carry on for a moment. So if you're going to go to the next slide, please. So these parts, so there's two enzymes. So an enzyme is just something that carries out an activity in a cell. I'm going to try and hold them up so you can see them. So there's the heart-shaped protease, and then there's the one that is really a bit like a donut. It has a hole in the middle, which is called replicase. If you just cut those out, as you can see on the picture, and then you can give them a little squeeze to make them a bit more three-dimensional, if you wish, so they can sit in your virus. So just give
I'm going
them
to mention the shape of them, of these.
Yeah. Oh, yeah. So these actually are based on. So scientists use something. There's technology that scientists use called X-ray crystallography, which is a sort of very high-tech way of looking at the shapes of proteins. And so these are actually based on the real shapes of the proteins. So we like to just sort of make it a bit more real in that sense. So those are, if you look at a picture online, that is what these proteins actually look like in reality. Okay, so next slide, please. So the genome, the genetic information inside the virus is in a form, it's called RNA, which is related to DNA, except it's a single strand rather than a double strand of DNA. So we've got these two parts to it, and we're just going to tape them together. So if you just want to tape them end to end, and the sequence, so you just do that. So that all the letters on there represent the genetic information, and these are actually based on, this is real sequence from a coronavirus, one of the early ones that was found in Wuhan. So this is from a patient, some of this sequence, and these letters are what allow the virus to make all of its proteins and do everything it does to make people ill. So if you just do that, so just pause for a moment.
Yeah. Well, I think we can pause the next one.
Okay.
Next slide, please.
Okay, so then just to show, so in the virus, the genome, the RNA genome, is actually protected by proteins, and they cause it to take on a kind of wiggly shape. So I just would like you to just curl and fold yours into a zigzag, your RNA. So if you just fold it up as I'm showing, as you can see more clearly on the picture than you probably can see me doing it, but then that will be a way of representing the protein. So the RNA is protected inside the cell, so it's not just sitting there on its own. But coronaviruses are actually one of the more advanced and complex viruses that exist, and so they have quite a lot more in them, but still not a massive amount of things inside them compared to our cells. Okay.
Should we pause for everyone to catch up with us and give us a thumbs up? Let us know how that you're with us. While we're waiting for you to do that, great, thank you for that. Just as Dominic touched on, there are different levels of complexities of viruses, and some viruses just have a protein coat which contains the genetic material, little particles, and some viruses have this envelope, and the envelope that you just made, that is derived from the membrane of the cell it infects. And those are the two basic types, and then there's the different types of genetic material within them. So yeah.
Okay. All right, we'll carry on. I'm not seeing that many thumbs. How are we doing? It's so hard to get feedback.
Can everyone write in the chat if they managed to create the virus up to this point, just to give us a thumbs up or an okay, yes?
Yeah, because we can hold on if you're not there yet. So, no. Okay. All right, we'll take that as good. Okay, can we have the next slide, please? Okay, so we're actually at the end of making the virus. So the final step is to just put all the parts inside. So you can just pop the RNA inside the virus, and that can just sit around the edge of the envelope, the membrane. And then the protease and the replicase can just, you know, sit inside. You can tape them in if you wish to keep them, glue them in if you want to keep it all held together. I'll try and show. There we go. There we go. So that is the virus made. And then we'll just, now I think we're just going to go through some of the things that it does, what these parts do, and obviously finish off your model if you're still going while we're doing that. So over to you, Melanie, I think.
Oh, sorry, this is, I'm not, can we have the next slide, please? And the next one, please. Okay, so we've labeled some of the parts of the virus here. And if we just talk about the sticky out bits on the surface of the envelope, so those are the orange and the purple, blue, and pink proteins. These proteins are specific to the virus and are involved in the virus attaching and getting into the cell. And some of the proteins are also involved in packaging the virus to get out of the cell again. The replicase and the protease are involved in dealing with the proteins that are made from the genetic material, which is shown as the RNA genome. So that's the genetic material in the virus. Next slide, please. Okay, what we're going to do is we're going to talk a little bit about testing and how the different parts of the coronavirus are targeted for different types of tests. Now, one of the things that happens when people are getting tested is they need to have a quick test. And when any new virus emerges, the first type of test that is developed is called a PCR test, and that stands for polymerase chain reaction. And it is a test for genetic material. You can do this test with a very, very small amount of genetic material. And what it looks for is it looks for if a person, it's looking for the virus particles to be present in a person. The limitation of this test is that it only tells you if a person has the virus right now. So it won't tell you if you've had the virus previously, and that is a limitation. But on the upside, it is a very quick test to do. It takes, once you have a swab and the swab goes to the lab, it takes a couple of hours to have the test done. All right, can we have the next slide, please? The other type of test you may have heard about is called an antibody test. And that is when blood is being tested to see if you have had an immune response against a virus. So it's to see if you have been infected by the virus. And it usually means that you will have immunity to a virus, because what antibodies do is they float around in your blood, and if they spot the virus that they are for, they will basically flag it up, stick to it, and flag it up to the rest of the immune system, who will come and destroy the virus particles, eat them, and also get rid of any infected cells in your body. So an antibody test will only work after you've been infected by the virus, and usually about a couple of weeks after you've been infected by the virus. It also takes a little bit longer to do. All right, can we have the next slide, please? Now, at the beginning, we talked about the problem with coronavirus being that some people are asymptomatic, meaning they don't have any symptoms of COVID-19. And the question is, why is that? Well, we have been, coronaviruses are not uncommon, and sometimes some coronaviruses out there in the world will just cause a cold, which isn't a problem. And our bodies will mount an immune response to them and produce antibodies to those coronaviruses, which will in turn also give you some protection from the current COVID-19 causing virus. So that's why some people might not be showing symptoms, because they might have antibodies which so-called cross-react with the coronavirus, and it helps the immune system mop up and destroy the virus. Now, This is the principle that has been used in vaccines since vaccines were invented. When Edward Jenner created the first vaccine for smallpox using a related virus, a cowpox, to create this immunity in people. So it's not an old new idea by any stretch of the imagination. I think that's all I want to say about that one. Any questions so far?
No questions in the chat yet. If anybody has got any, please feel free to pop them in the chat. Melanie and Dominic will be happy to answer them for you.
Okay.
Have you made your models?
Yes, what I am going to say is if any of the schools want to email me their models, a picture of their models, take a photo, email me a picture, and we can put them on our website. And I'm sure if I can, Melanie and Dominic would like to see them as well, so I can.
I'd love to.
That'd be great.
Oh, here's a good question. Why do some people seem to get coronavirus more than once?
Good question.
That is a good question. Well, there could be a few reasons for that. For example, it could be that the virus does change and mutate. And sometimes some of these changes in the genetic material cause changes in the surface proteins, which mean that your immune system can't fight it as well anymore. Now, this is not also something that's unique to coronavirus. This happens every year with flu. Every year, the flu vaccine is towards a different version of the flu vaccine because it changes every year. So it could be that is the reason that it is maybe changing more quickly and some people are getting infected twice.
Yeah, I mean, in terms, just going back to the model and just thinking about the genetic information in there, because the enzyme replicase copies it, and when it makes copies, it does good quality copies for the first part of the genome, but gets worse as it goes on. And the bits that it's worse at are the spike protein, and these are, which is how it gets into the cells, and the things called accessory proteins, and these are how it avoids the immune system. So those are the ones that are changing most frequently, and I'm sure that is what's happening with the people getting it twice. There's a few other questions now.
We have a question. How long does it take for the virus to leave the body? In all honesty, I don't know the answer to that.
I guess if you get over it naturally, I suppose it's probably a week or 7 days, like a normal immune reaction, isn't it?
Usually, to mount your immune response to a virus, it takes between 7 and 10 days to get antibodies to it. And then after that, the cellular part of the immune system will be clearing up the mess, as it were. But I can't give you...
Obviously, people end up in hospital very ill with it, haven't been able to get rid of it on their own without support. So it can take a lot longer. And then there's this whole thing about long COVID, which I don't think is really understood yet what that means. How about the question? So there's one question about how long does it stay on surfaces or in the air? I mean, I read a paper, there was a report this week actually about saying it could last for, was it quite a long time, isn't it? It was many, many days. And it's around, it's just the question is often it's not clear whether it's really infectious and how long it's infectious for, and it's quite hard to study that. I think the scientists try and make it as realistic as possible when they do these tests. I think they were saying as long as 28 days recently. So whether that is still infectious to a person in a normal environment, if it's on a cardboard box that you've got from Amazon, or, you know, an envelope through your door or something else, I don't think people really know. It's quite tricky to research that, I think. Yeah.
And I think the other thing as well is the method in which the virus is being tested for still being present will also possibly give you the wrong idea. For example, if you're using a DNA test, a PCR test, you can detect minute amounts, which doesn't necessarily even mean the whole virus is there. So it's difficult to say.
Okay, next question. Is there a step-by-step guide? I mean, there is
everyone got the...
Yeah, with the templates, there is a printout that you can print it out or email it of the slides, which shows what to do, or else it's the PowerPoint.
We will be sharing the recording, so that will be put on the website as well, so they will be able to look at it again and view it that way.
Great.
Okay. Are only young people getting long COVID? I don't know I don't know the answer to that. It seems to be that young people get a much milder thing.
Yeah, in general they do. I mean, most young people hardly get anything, and this is the problem with it. It's unusual in that it's very mild for most people, but then for other people it's very serious. And this is the kind of, and the reason it spreads so easily is because the people who have it don't know they have it most of the time, and so they can spread it without me knowing they're doing so. And then of course it unfortunately can reach someone who's susceptible to it, who can end up in hospital with it. So it's quite a problem.
That's one of the big questions, is what is the difference between people who are getting it very badly and people who are asymptomatic? And there is, you know, when I think about biology, there is so many layers and steps from a molecular interaction to a whole organism. And there's a lot of mystery in there. We don't really know a lot about how it works. And there's so many things at play. So it's hard to, you can't predict it. And I think if you could predict it, that would be great. And, you know, the extent of the underlying health conditions is an attempt at predicting that. But I don't think we can predict it any further than that. Why are so many people asymptomatic?
I think, how many?
We were talking about, no, we did only young people getting COVID. And asymptomatic, did we do that already?
I think so.
Okay. How many viruses are there? Oh,
Lots. There's many. I mean, viruses are interesting because people don't really know whether they were a very primitive life form that is carried on, like something that was alive, as it were, in the, you know, primordial soup billions of years ago, or whether they're parasites in that they've lost things, so they've become simplified and they only are able to survive by infecting cells. But there's been viruses around as long as there's been life on Earth, so there are millions, there's probably certainly millions of viruses, I should think.
I mean, kind of in reference to the question above about why are so many people asymptomatic, there's lots of types of coronaviruses, you know, those alone. And so that's why, you know, you can get those and not get that sick and have an immune response, and then therefore, you know, that helps you fight any infections from it. And I think that's the reason really why so many people are. But there are so many, viruses are fascinating. As I said before, there are so many types that some have an envelope, some don't. Some have a DNA genome, some have an RNA genome. There's so many different types of them, causing many, many different things, and they'll infect different types of cells. So, you know, really an interesting area.
Good question. How long will it be around? How long will COVID be around? I mean, who knows? Maybe several years. I mean, I don't know. I mean, that's the big question, really, and it's an excellent question that has been asked there. Certainly for another, I don't know, probably longer than we want, that's for sure. A year, maybe several years, maybe less. Depends if a vaccine's found, I suppose, or a vaccine that people can get hold of.
I think that leads us on. We'll come back to this next question in a minute, but if we could go to the next slide, please. Let's talk about vaccines for a minute, because this is the big question. I mean, we're getting, you know, how long does a pandemic normally last like this? Normally last, we don't really know the answer to that. Because what we're trying to do now, scientists are trying to produce a vaccine to help protect us and vulnerable people from the infection with coronavirus. And there's 3 different ways in which scientists are designing vaccines. So a vaccine is when you inject a person with some form or part of a virus so that you get your own immune system to make antibodies against that virus, so that if you are infected with the real thing, those antibodies are there, they'll mop up the virus and target your immune system to destroy any of them already present without making you ill. And we, as babies, are all given lots of vaccines for illnesses, and they work very well. Now, the targets for vaccines for coronavirus, one of them are looking at the surface protein. So preparing something like the spike protein as a purified form of spike protein that could then be injected into people. And that would then make your immune system make antibodies, which would recognize that spike protein if you were infected by the real coronavirus, flagging up the virus and any infected cells to get your immune system to fight it. That's one way. Another way is using so-called live attenuated virus particles. So those are coronavirus particles that are purified in the lab and changed in a way so they don't make you sick. those are then injected into you. The idea being that it's not just one protein, that you could get lots of different antibodies against lots of different proteins, maybe give you a stronger immune response, and to fight the infection if you got infected with a natural wild type virus. Another virus strategy for vaccines is to create a vaccine where you have a virus like one called adenovirus, which causes the common cold. Those are used widely in labs, and you can engineer those virus particles to have some of the genetic material of the coronavirus, which will then produce these proteins on the surface, and then you can get an immune response to that. These all take a long time to develop. They take many years usually to develop. One of the sort of newest technologies to try and make vaccines is using the genetic material itself, the DNA or RNA for a vaccine. And the idea with that is that you would inject a person with the genetic material only of the virus, and then your own cells would make the proteins and then get your immune system to produce antibodies against those foreign proteins. Now, that technology hasn't been successful in humans yet. I've read there is a horse vaccine for Zika virus using that technology, but that's where that one is. So there's different companies, many companies around the world trying to make these vaccines using these different methods. And we're waiting to see what happens. Now, as I said before, vaccines usually take years to develop, and it's the speed at which these are going. You know, we've already heard about some virus or vaccine trials being stopped because people are getting ill from the vaccine, and you have to investigate those sorts of things to see what it is that's causing it, because it's not a natural thing to happen in this way. So we have to just watch this space.
Okay.
So
should we just pick up a couple
yeah.
more questions? We're getting
Okay.
One question is about why are some groups of individuals more at risk, and there's been a lot of clearly there's some issues with certain ethnic groups and so on are more susceptible or getting more seriously ill from the virus, and I don't think anyone knows why that is at the moment. It's just because the problem is the virus hasn't been around that long, and so to sort of research these topics I think is going to take quite a long time. But clearly there are differences in different groups, and older people and
People with asthma,
with certain
like that,
medical conditions and so on. I think unfortunately we'll probably almost have to wait till it's over to find out the answers to those questions. That was a nice question about CRISPR. So could you use CRISPR to attack COVID-19? So CRISPR, for anyone who hasn't heard, last week actually, the Nobel Prize was given to two scientists who have developed a technique which is called CRISPR, which is a way of genetically engineering genomes, and it's very powerful, and they've got the Nobel Prize for it very quickly. And these two women working, one's in, I think that one's in France and one's in the US, and they've worked on this project. And this technology, you can use it in lots of different ways. I've not seen anyone suggesting the use of it to develop a way of attacking the virus directly, but certainly CRISPR will be used in the development of vaccines and medicines to work out how to destroy coronavirus.
Another question is, can pets get the virus? I've only heard this in
no, there have
news,
been cases, definitely. Yeah, dogs and cats have definitely got it. Because it's crossed between species, I mean, you can see it came undoubtedly from a bat, it seems, via a pangolin, which is another animal in China. And this type of virus will cross between species barriers, but it doesn't necessarily mean it will make pets ill, and they may just be carriers, or they may just get it at a very low level and not pass it on. Again, I think this is just something people are researching. I mean, normally these things take years to find out. And of course, this is happening on sort of fast forward because the virus is so problematic that people are trying to understand these things very quickly, but some of them just take quite a long time to understand. But yes, pets can definitely get it, but how often, I don't think we can answer.
Okay.
Right.
Are there any other questions?
There's one other question from Emma McMorrow. How long does a pandemic like this normally last?
Oh, sorry.
How long does a pandemic like this normally last?
A few. Well, I was talking to a doctor who's a surgeon the other day, and he's completely convinced 3 years. I mean, this is what has happened in the past. I mean, I don't know. I mean, probably more than, it's certainly going to go on beyond March next year, isn't it? It can't see it disappearing before at least next summer, and probably beyond that, really. I mean, I think I don't know, people have different views on whether we need to learn to live with it and adapt around it, or whether we kind of keep doing lockdowns, and it depends on people's view on those things. I mean, the politicians are kind of tending towards the lockdowns at the moment, but maybe you know, maybe we will have to learn to live with it in some way as time goes on if it doesn't go away and there isn't a vaccine produced quickly. I guess time will tell.
We have another question.
Oh.
Will there be a second wave? I think we're in it.
We're riding it right now. Yeah.
I think one thing to be aware of, when we're seeing all these graphs that go up and down giving us different waves, something that's important to remember is the number of tests that have been done back in March during the lockdown was far fewer than are being done now. So even though we seemingly have more people testing positive now, it's because there's a lot more tests being done compared to in lockdown, where only people in hospitals were really getting tested. So there was probably a lot more people infected then, but we just didn't know. It's one of those things, why, you know, about critical thinking when you're reading these reports and seeing the numbers, just to, you know, really question what are the numbers telling us and where are they coming from.
And obviously as time goes on, more people have had it, and there have been a few, I mean, I think there's been half a dozen cases where people have got it more than once, but most people don't seem to. So as time goes on, millions and millions of people become immune to it, and so that will naturally kind of reduce the success of the virus in spreading, because there's far fewer people to infect. So that will happen over a long period of time. Okay,
we have another question. How can we get the antibody test? I think you can get the antibody test done just in pharmacies. I think you can, but the thing about that to bear in mind is sometimes in the news I've been seeing people saying that the antibodies don't last forever. I don't know if that's true. That's kind of goes against all of the theory and all the reasons why we vaccinate people to begin with. So that's all I know I can say about that.
We do make a model of the antibody
we
test.
Do, that's true.
Here's the next slide. Oh, we're jumping.
Oh, yeah.
Oh, right. We've got to the last slide.
Okay. I think that's all we had to say, unless there's any other questions.
No, I don't think there's any other questions. So thank you, Melanie and Dominic, for that. That was a really good interactive presentation. So the screen will be stop sharing now. And once we are all done, like I've said before, if you want to take photos of your presentation of your models, please send them to me. It'd be great to see them, and we will put them on the website if you're happy for us to do that. There's going to be a feedback link that Helen will put in the chat just in a second. If schools can give us our feedback on this session, it would be really, really useful and much appreciated. So that will go in the chat shortly. So,
I think we'd just like to say thank you to everybody for joining us today. We've really enjoyed doing this and really enjoyed listening to your questions. So
hope you enjoy your virus, probably isn't the right to put it, but you know what I mean. Enjoy your model.
Yeah, and I hope it's shed some light on what's going on scientifically behind this crazy time we're living in.
Lovely. Thank you very much.
Okay.
Thank you both. I've put the link in the chat, so if you could put all your feedback down, that would be great for the schools. And if we can ask Melanie and Dominic to stay on the call until everybody's left, that would be fantastic. But thank you again for joining us today.
Thank you.
Thank you.
Thank you all.
Cheers, thanks.